Hi, my name is Madison Oliver, and I'll be presenting my research about how distance affects resin choices for our native solitary bees. So why is it important to study native solitary bees? So both honeybees and native bees have seen a significant decline in population. However, people mostly or only know about honeybees. Could be because we only see honeybees in the media, or because honeybees produce a product. Of the studies of their decline, much more attention is given to the study of honeybees than native bees. Again, this could be because honeybees produce a product. Resin is a primary nest component for some bees. However, there has been little attention given to the study of resin and resin usage by bees. This doesn't mean there has been no studies, but there has not been a lot about their choices and their preferences. But it needs to be further looked into. So what is a native solitary bee's life cycle? So native solitary bees have a very different life cycle than what we think of honeybees. Females will mate with males, and then they will find places to build their nests. Usually, when we think of resin bees, they will find holes and they will lay their eggs on pollen that they collect. They will then create like a cell, and they will create several of these cells and then cap off the nest. They will not protect this nest. They will leave to potentially make another. After about 6 weeks, the adult bees die. The eggs laid will overwinter and will emerge in the spring or summer as an adult bee, start the life cycle again. How do native solitary bees use resin? Ways and choices around how native solitary bees use resin is not studied in depth, though there is some literature. Well, we know that native solitary bees use resin to make cells of their nests, like as shown here. We don't know if or why they are making specific resin choices. So there are cases of some solitary resinous bees making specific resin choices. We don't know why they are making those choices, and we don't know if all resin bees are making those choices. We also don't know why some resinous bees choose to mix resources in with the resins they use. One thought is that it might be due to the scarcity of resin in their area. Furthermore, if they are making specific resin choices, we don't know why they are making those choices. So my observations. In Minnesota, we have conifers that produce resin internally. That would be in their trunk, and temperate angiosperms that produce resin at their leaf tips. Some resins are more readily available than others, i.e. some conifers, when they are wounded, only will produce a lot of resin, while others only produce a little. And some places, conifers and trees in general are wounded a lot, while in other places they are not wounded as much. And in certain angiosperms at certain times of the year produce more resin than other times of the year. Honeybees discriminatorily search for balsam poplar and eastern cottonwood resins. And some Myglochilidae have also been found to be selective with their resin choices. So this was a paper about Myglochilidae in Arizona. They were found to prefer brittle-bush, which is a resinous plant found in the desert. But again, we don't know why they prefer that resin. And we don't know if other megachile are also making those kinds of selective choices, or if it's just scarcity factor. So my question, are native bees selective with their resin choices? Hypothesis. Native bees will use the closest available acceptable resin source. This means that they will travel the least distance to get their nest materials. So they're going to use the least amount of energy to get their nest materials. They're not going to want to fly as far. And I think they'd even want to fly less far than honeybees. But that's just a personal thought, because they are smaller than honeybees. So experiment number 1. My prediction was, if boxes are located on different species of resinous trees, then we would see approximately the same amount of nesting across all species of trees. My variables were, my independent variable was nesting, and my dependent variable was the nest box location or the species of tree. So in this experiment, we chose several different locations, and we had 21 boxes, and we strapped to 3 different individuals of 7 species. They were observed over several weeks. And I have my results here. So here is a table showing the appearance of nests by week. This is all nests, so this includes, we saw nests made from wood, nests made from soil, and nests made from resin. As you can see, we had a total of 32, which I think is decent. What I think is interesting is that it almost kind of comes in, the bees nest in kind of waves. So as you can see, we have a big jump in this first week here of the 27th. And then we have 2 and 5, and that's okay. But then we have another week of 7, which I think is significant because that's almost as much as the first week. So that's the week of the 12th. So not quite a month later, couple weeks later about. And then we've got another couple weeks in between, and then we have another spike. So it is coming and going in waves, which I think is very interesting. I don't quite know what that means, and I think that would need to be researched further. Going off of that, I decided to take just the resin nests from that table. So as you can see, we had a total of 16 resin nests. And they really go with that wave that I was talking about. So we have 6 in that first week, we have 5 in that week of the 12th, and we have 4 on the week of the 1st of August, which do make up the majority of the resin nests that we did end up seeing. I don't know if that if these are significant weeks for them, or if they just have this kind of couple weeks break in between where they need to gather materials. which it would be my guess. And as for my prediction, I decided to make a table of resinous nests per species of tree. Now, as a caveat for this, there were several boxes that were taken out of my data that did have resin nests by the end of my observation. So had they not been taken out of my data, maybe things would have been different. However, stuff was done to them that made them not reliable. So as you can see, all across the board here, we have about 1 to 2, except for in the white pine to white spruce, we have 5 and 6, which does seem significant. However, I do think that the areas where these white pine and white spruce were better as far as floral resources, less human activity, sunny areas, etc. And I will talk more about that as we go on. So discussion. No nests were found in column 1, which had the biggest holes, which I think is significant. Conversely, most nests were found in column 3, which had the smallest holes, and all resin nests were found in column 3. Big, like all resin nests, they like really, really, really small holes for some reason. I think because they are small bees. Seeding the box with old nests did not seem to be useful. As we found that seeding the boxes, as we found that the boxes that were seeded had an 81% chance of having a nest, and the boxes that didn't had an 80% chance of having a nest. It is a possibility that the bees that came out of the wood nest blocks nested again in the wood nest blocks and not in our bee boxes. We seeded them with a wood nest block that had nests in it from the previous year. So it is a possibility that these bees emerged from the nest and then nested again in the same wood block. Floral resources being near human activity and the location of the tree seem to matter a lot, which introduces a lot of variables. And this is something I was saying before. It's hard to control that when you're just putting bee boxes out into the world. But that's also where the bees are. So my prediction was not supported by this experiment. However, I do think there was some flaws in our setup. Some of the trees were in much better locations. They had more floral resources. They had less human activity. They were sheltered but still sunny. And 3 of the boxes were taken down. I didn't include their data, but I did record it, and there were several resident nests in those boxes by the end of the season. This could have made a difference in the data. But then again, they were taken down from their trees, and we don't know if the bees found the box when they were taken down or when they were still on the trees. So we don't want to use that data. So experiment 2. My prediction was if native bees nest in our bee boxes, then they will choose resin from the tree that the bee box is attached to. So my independent variable is the nest cap composition, and my dependent variable is the distance from resinous tree. So my experiment number 2, I made boxes for Cedar Creek by drilling some small holes and some small planks. I collected resin from 7 types of resinous trees on the St. Paul campus, as well as collecting from ponderosa pine at Cedar Creek. And I used a needle to collect resin from the resin caps that were found in the boxes or the planks at Cedar Creek. The resin samples were mixed with 70% ethanol, filtered, and prepared for analyzing. These are all my resin samples. As you can see, you can get quite a different color from them. And while most of the resin caps I saw on campus were white resins, so it would be hard to tell what tree they came from, I did see some red resin caps, which would likely be from balsam poplar. I did want to make a note of that while we were here, so it's easy to see. Continuing on, we used LCMS to analyze the samples to determine the source of the resin caps. In simplest terms, LCMS is a machine used to analyze and compare liquid substances. LCMS separates compounds over time based on polarity and gives a reading called a chromatograph. The compounds that appear later in the chromatograph are more non-polar, while the compounds that appear earlier in the chromatograph are more polar. We can see the mass of these compounds. However, this does not mean we can identify them. This does give us a way to compare these substances, which I will show you. So here we have a chromatograph of my resin caps and the ponderosa pine resin that I collected. Sorry, I'm going to move my head here just so you can see the end here. So as you can see, these peaks are lining up, and that is very significant because that means that they had similar compounds exiting the column of the LCMS at the same time. Which means they are likely the same substance, but what would it look like if they were something completely different? Well, here's one of my resin caps versus some of my resin samples, my other resin samples. So my balsam poplar, eastern cottonwood, and white pine. And as you can see, they peak in completely different places. They look completely different. They don't have a lot of similarities, which means most of their compounds are different. It's likely that what the resin cap C1 is made out of is not balsam poplar, eastern cottonwood, or white pine. So the resin caps I collected from Cedar Creek were most likely from ponderosa pine. We did lose one resin cap sample in the process of filtering it. It cannot be assumed to be ponderosa pine, but it is worth noting that there was one more. My prediction did seem to be supported. However, I think that more experiments should be done to further investigate resin bees and their choices. I was only able to strap boxes to a small area at Cedar Creek, the only resinous trees in that area were ponderosa pine. So I do think that the resin there was it was a scarce resource in a way. So what's next? I think to repeat experiment 2 again with a wider variety of resinous trees in the area would be good. I also think to try a different experiment with resin available, but to modify the floral resources, the human activity, or the location, make it sunny or shady, and see how that affects nesting. Of course, only modify one of those variables at a time, just to see which one of those has a bigger effect on how bees nest. If one of those has a bigger effect, or if all of them have an equal effect. My acknowledgements. I'd like to thank you all. Thank you, Professor Wilson. And that's my presentation.